I'm honored to have Dr. James White of Rutgers University, who is a full professor at the Department of Biology at Rutgers, and he's a plant microbiologist, and he has authored about 300 manuscripts, scientific journals, and published 8 books. And he has been recipient of numerous awards, including the Alexopoulos Research Prize from the Mycological Society of America. And I'm honored again to have him to present a very important concept in the interaction of the microorganisms with plants, and how microorganisms assist in the nutrient uptake of by plants, and how plants basically form the microbes. Jim, I'd like to turn the time to you, and again, welcome aboard.
Thank you, Moj. I really appreciate you inviting me to do this, and all of your guidance and counsel over these years. I really appreciate knowing you. Really appreciate it. So I will go ahead and share my screen and begin to. Okay, so how does that look? Does that look pretty good?
Yep, it's coming through. It's, if you like to maximize it, then we'll have to.
Yep, that's what I'm going to do. I'm going to see if I can get that. There we go. There we go. So what I'm going to talk about are how plants use soil microbes, soil microbiology, and specifically a couple of different concepts, kind of biological processes. One has come to be called the Rhizophagy Cycle or Rhizophagy process. This is simply the process whereby plant roots manage, farm microbes, and the other is actually endophytism, and how endophytism works in plants, and how all of this works together to create healthy plants and healthy agricultural system. So I'm going to touch on really comparing what I call biological agriculture to conventional agriculture. I could say conventional agriculture, I could say purely chemical agriculture, meaning we manage plants with chemistry, versus biological agriculture. And that is, the biological agriculture is emphasizing more of the biology, the microbiological aspects of plant cultivation. That is where we use microbes to cultivate plants, and we support soil microbiology. And I'm going to talk about what endophytes are, and these are basically microbes that go inside plants and do not cause disease, as opposed to pathogens, right, which go into plants and cause disease. So endophytes are non-pathogenic. I'm going to talk about how plants will vector microbes on their seeds, and how that's important for healthy agriculture, healthy plants, because the plants actually need the microbes coming out of the seeds. They also get microbes out of the soil, but they carry some in their seeds, and so we have to have microbes on seeds. I'm going to explain the biology of the rhizophagy cycle, the rhizophagy eating cycle, where plants internalize microbes from the soil into their cells at their root tips, and then they extract nutrients from those microbes inside their roots. And I'm going to talk about a process that's come to be called habitat adaptive symbiosis, and that is where plants use microbes to adapt them to their environments. So if it's a highly saline environment, they'll use microbes that make the plant more resistant to salt. Okay? And whatever the stress is, plants can absorb microbes to assist in their adaptation to that environment. Habitat adaptive symbiosis. And finally, I'm going to talk about how plant microbes and soil microbes in general can affect pathogens in the soil and cause them to be non-pathogenic. So basically, where soil microbes, plant microbes can modulate fungal diseases and reduce the need for pesticides, fungicides, for example. So just to contrast the conventional agriculture to biological agriculture. Conventional agriculture is what we mostly do, where we manage crops using chemistry, using fungicides and herbicides and chemical fertilizers. And we basically, in this process, we anticipate what plants need, and we put the chemicals there. We put whatever we think the plants needs. We grow that. We manage the crop. And of course, at the end of it, we have products, and those products are good edible food, but they're not actually as rich in nutrients and antioxidants and so forth, other compounds that biological agriculture yields, or that that plants grown with microbiology yield. Actually, the crops that come from microbiological cultivated plants are richer in antioxidants and phenolics and nutrients, and one estimate is like they're 30% higher in nutrients. So depending on the study. Okay, in biological agriculture, we basically either use microbes directly, putting them on to plants, things like compost washes, also cultivating soils so that they're rich in microbiology, using cover crops, for example, to increase the microbes in the soil. So it's a different approach. Is it possible to fuse the two? Yes, all the evidence that I've seen says that we can fuse these two methods, reducing the chemical inputs, increasing the biological inputs. That can work. We just have to explore precisely to what extent we could fuse those two, but evidence is it can work. With chemical agriculture, we basically are doing this. We're putting chemicals onto plants, and I'm not a great artist. You can see I have a plant cell here. This is the plant cell, my rendition of a plant cell. I put a little funnel there. Basically, with chemical agriculture, we're trying to fertilize by getting the nutrients into those plant cells, into that cell. But the fact is that few nutrients actually go into the cell. This chemical agriculture, in terms of fertilizers, is really only 30 to 40% efficient. That means only 30 to 40% of the chemicals that we put on plants actually go into plants, actually end up in the plants. Most of it ends up in the environment. We waste the chemistry and we do damage to the environment and do damage to the earth. This is the problem. So a lot of people are trying to figure out how we can get away from that. Biological agriculture is different. It's very different. And a pure biological agriculture, basically nutrients we use microbes, and those microbes go into plant cells, and I'll show how that happens. They're absorbed into plant cells, and then the plant will extract nutrients from those microbes in the cells. And basically they oxidize nutrients out of the microbes, and I'll show how that happens. But this process, the biological, the endogenous nutrient supply using endophytes, using microbes, it is what wild plants do. No plant in nature needs fertilizer, needs our chemical fertilizers. The chemical fertilizers mess up the systems when we put too much on. Okay, so the plants don't need it. And the other thing about this biological agriculture, this endogenous bacterial fertilization system, is that it's 100% efficient. There's no loss, or I could say very little loss. We know that some plants that are doing this have a little bit of nutrients that leak out from around the roots, but it's very, very little. This is basically an efficient system. We just have to figure out how to actually use it, how to do it, how to make things work. And that's going to take all of us to do that. The growers and companies and academicians. It's going to take everyone to figure out how to do this. But endophytes are basically the focus here, because these microbes that provide nutrients, many of them are endophytes. I mean, some may be mycorrhizae, we know about that, but many of these microbes go into the plant cells, into their tissues, and then provide nutrients to the plant. Endophyte is any microbe that doesn't cause disease, but still goes into the plant and does not cause disease. This is a fungal endophyte, and a lot of people are familiar with fungal endophytes in grasses, like a tall fescue, a fungus that goes inside the grass, inside the leaves and the stems and the seeds of the grass. And you can see this blue line, that's the fungus inside the plant. Fungal endophyte. Okay, this is actually a bacterial endophyte, and this is you can see the blue things in here. These are cells of a bacterium called micrococcus that produces these clusters of cells. So there's tetrads, essentially, micrococcus luteus. But we took the bacterium out of a tomato, and then we put it into a weed, a plant called curly dock. Rumex crispus is the scientific name, but we just took the seed of the Rumex crispus. We cleaned off the microbes of it. We sterilized it as much as we can by cleaning it off, and then we put this bacterium that we got out of this endophyte from tomato on it. And then when the plant germinated, when the seed germinated and the root came out, it took in these microbes into the cells. And you can see them in there now, and you can see they're in the cells. See the cell walls there. They're actually inside the cells inside. So what are these microbes? These microbes are soil microbes. This is where plants get these microbes from. That's why we want to build healthy, rich, microbial rich soils, so the plant can get them, absorb them into their tissues and use them. These microbes also are seed vectored. So healthy plants growing in healthy soils will get the microbes, and they'll put some of those microbes onto seeds. And then those microbes on the seeds will be there when the seed falls to soil. If there aren't immediately microbes there, the seed already has microbes. The seedling will already have these microbes that are seed vectored. So we have to be careful when we treat seeds. We need not to treat seeds with anything that will kill the microbiology on the seeds. And of course, some people feel like or think that part of the strategy is that we need to kill the microbes on the seeds because some of them are pathogens. And that is true. There could be pathogens there. But the fact is, from the experiments that we've done, the more microbes you have on the seeds, the more the diverse the community, the less problematic any of these fungal diseases like fusarium will be. And I'll talk about that later on. I'll show some experiments about that. But in our work, we can put more microbes on seeds to control the pathogens. So we actually need not less microbes on seeds, but more microbes on seeds. Okay, this is just a grass seed germinating. And this was, by the way, this is my student who did some of this work on grasses. But you can see there's where the arrows are, you can see the yellow. That's the bacterium coming off of those seeds. The seeds usually have some kind, or many seeds have some kind of accessory tissues on them that carry microbes. So this grass seed here has paleas and lemmas that is essentially dead tissue on these seeds, but microbes are carried there. You remove those and the plant is not going to have as many microbes. Now, there are some microbes carried inside the seed around the embryo, and we can see them in there, and those we cannot sterilize the seed to remove. So some of them will go inside, some of them go on the surface of seeds. Consider tomato, the seeds in tomato. We generally think that these plants, tomato, especially in the fruits, they're sterile. That's far from true. In fact, around the ovaries, if we look at these seeds here, around the seeds, there are microbes in the cells. and when the tomato seeds are produced, they'll have covering the little accessory tissues that are hanging on those tomato seeds actually have microbes on them, and also inside those seeds. We can cut the seed and we can look in the embryo and we can see the biofilms around those embryos. They're different microbes. There's not one microbe. Plants have different microbes depending on where they're growing, perhaps exactly what the environment is that they need to adapt to. They'll get different microbes. So anyways, and I already made this point. We have to be careful with seed sterilization and how we treat seeds to preserve the healthy microbiology on those seeds. I'm going to go into now this discussion of the rhizophagy process that happens in roots, and this is rhizo for root, phagy, eating. Rhizo root phagy eating process. Okay, this is where plants are absorbing microbes from the soil, and they're using them for development, so the plant can develop properly, and I'll explain how that happens. And they're using them for nutrients, and I'll explain how that happens. I've put here at the bottom 3 key papers. These happen to be from my lab about the rhizophagy process, and basically data that supports what I'm about to tell you and what I'm about to explain to you. So you could go to these papers and you could get more details. And these are all peer-reviewed. They're all open access, so you can get to them. They're there. So I encourage you to look at that for more of the data to explain how this works. And this actually, I should explain, in my lab, we didn't discover rhizophagy. This was actually discovered in Australia in 2010, or leading up to 2010. A publication was made in 2010. The publication is here at the bottom. And basically it was a paper published in PLOS One, very well done. It's, again, PLOS One is open, as you likely already know, is an open access journal. But it is titled "Turning the Table: Plants Consume Microbes as a Source of Nutrients." Okay, as opposed to microbes consume plants as a source of nutrients. And these are the couple of the investigators that did this work. There was a whole team there, and I don't have them all listed. They did this work, they published it, and then they later in a chapter that they published a couple of years later, I think it was 2012, 2013, something like that, they denominated the process rhizophagy, root eating. So they called it that, rhizophagy. What you can see here is this is actually tables. This is from a table of figures from their paper. And if you look down here at E, you can see these root hairs. And I think this is tomato probably here. And then you see these little green dots in there. What they did, these investigators, they labeled a yeast, and they labeled the bacterium, an E. coli, and they labeled the yeast, the Saccharomyces, simple yeast, with green fluorescent protein, and then they put it on plants. And then they used confocal microscopy to track those microbes through the plant, into the roots. And they showed that they went into the root hairs, into the root cells, and into the root hairs. And then later, the fluorescence disappeared. So these investigators concluded that plants are now consuming these microbes. And these investigators at the in Queensland, Australia, I think at the University of Queensland. I think that's what it's called. But they're still down there doing their work, doing their thing. So what we have found is that it's not only bacteria and yeasts or fungi that plants can absorb into their roots, into their cells, but also algae and perhaps other soil microbes. This is more recently, we looked at a chlorella about a year ago, and this actually shows this chlorella. It's a green alga. A very interesting thing, because you can see bacteria around these chlorella cells. And these chlorella also have their own endophytes. And it's curious, the plants will absorb these chlorella, this green algae, into their tissues with their endophytes, and then they will degrade the algae, and they'll get the bacterial endophytes from the chlorella. So it's in consuming the green alga, the plant gets their bacteria. This actually below, you see the picture, that's a root hair of a plant. And you can see the chlorella inside. That's the green alga there. You can see it's losing its chlorophyll. It's losing its chlorophyll because the plant is exposing these microbes to superoxide, which is a form of reactive oxygen, and it's degrading the chlorophyll. It's causing the chlorophyll. These algae are losing their chlorophyll, but also these bacteria are coming out of these algae. And you can see the little dots around it. Those are the bacteria coming out of that green alga. If you look at the yeast, you can see here, actually, this is a clover root hair, and you can see the yeasts coming out of the root hair tip, and this is how they emerge. This is what happens in the rhizophagy process, in the rhizophagy cycle, and I'll show you that. I'll show you the details of that. But inside, you can see these little dots here. Inside, those little dots are actually protoplast phases of these yeasts, of the yeast. And what happens is the microbes present inside, they're wall-less. They don't have walls. They have only protoplasts, and they can be little tiny like that. But when they're ejected, they begin to grow because the plant puts exudates out with them. And you can see here, if you look at the smallest, you look closer to the hair, you see the small yeast cells, and then you see older ones are bigger. That's because the plant is actually putting exudates out there through the root hair, allowing these microbes to recover and become soil competent again. The same thing happens. This is bacteria here, and you can see the root hair tip, and you see the bacterial protoplasts inside. Now, you may wonder why we haven't seen this before, because this was only discovered 2010. Why was it discovered in 2010? Well, why so late? Why didn't people see it before? Because these microbes, you don't know they're microbes without their cell walls. They look like vesicles. In fact, that's what people have called them. They call them vesicles. And sometimes they call them, interestingly, extra cytoplasmic vesicles, otherwise known as microbes being ejected from root hair tips. But they are microbes. And the reason we can see them so well is because we use stains called reactive oxygen stains that will turn color when you have reactive oxygen, like hydrogen peroxide, or you have superoxide that are produced by the plant onto the microbes. And because there's this interaction, because the plant is putting reactive oxygen onto the microbes, with these stains, then we could visualize the microbes there, because the plant isn't putting them on non-microbes. They're putting only on these microbes that are inside the root cells. So what we have shown is that it actually is a cyclic process. And this is a diagram that shows what we call the rhizophagy cycle. And basically, with the rhizophagy cycle, we have soil microbes being attracted to plant root tips via the exudates. In other words, at the root tips, plants secrete exudates. And these exudates are carbohydrates. Sometimes there may be some oligopeptides in there. There may be some organic acids and other small molecules. I've heard there's also vitamins sometimes that go out from these exudates. But those exudates are attractants to soil microbes. And microbes will follow the chain, the trail of exudates back to these root tips. And then the plant will cultivate those microbes at the root tip. And then at these root tips, the plant will absorb some of those microbes into the root cells. And then as soon as they're absorbed, the plant cell will detect the microbes because the microbes are secreting ethylene, which is a plant hormone that causes the plant to actually secrete superoxide. And then the plant cell secretes superoxide onto that microbe that just entered the cells. What that does is it will oxidize off the cell walls off of those microbes, and in doing so, acquire nutrients that were on those cell walls. And I'll explain more about that. But the plant will get the nutrients, absorb the microbes, oxidize the bacteria or yeasts or green algae, whatever they are that they're absorbing, and then process those microbes that are now inside the cells, the wall-less protoplasts of the microbes, process them, replicate them, increase their numbers, and then the microbe protoplasts will actually go into the root hair initials. So the unexpanded root hairs, the little initials, which are just little bumps on roots, they accumulate these microbes in them, and then those microbes in bulk that are in these root hair initials will secrete growth hormone. Ethylene is probably the main one that causes root hairs then to grow. And then you have growth spurts happening, repetitive growth spurts happening of the root hairs in response to presence of the bacteria in those root hairs. And what happens is that as the growth spurt happens, some of the microbes are ejected out of the tip back to the soil. So they go back to the soil and they can acquire more nutrients and attracted back later. And what I want to show you here with this image is this is an actual image of a root tip. You can see these root hairs out here. You see the little bumps at the tip. Those are bacteria that have been ejected out of these growing root hairs. And these bacteria, every time these root hairs grow, triggered by the bacteria, then there's a growth spurt and an ejection of microbes back to the soil. And then you'll have a buildup in these hairs of microbes in the root hairs. And then they'll accumulate at the tip. And I'll show you how they do that. Inside the tip, they'll produce hormones and they cause another growth spurt. And then you have an ejection again of those microbes at the tip. I'll show you that. So that's a nut in a nutshell how the rhizophagy cycle works, but I'll show you the details of that. Now, I will point out, why are they able to go in at the root tip? They're able to go in because these root cells at the root tip, this is where the root cells are dividing. This is where they're still growing. This is where the cell walls are plastic and elastic. This is where they can go in. This is where the plant can get them in, can take them in. Why are they coming out at the tip right there? Because that's the other place that the root cells, the root tip, the root hair tip is plastic. It's not hard. It's soft, very soft there. Okay, so you see an entry way and you see an exit way. And this is how it works. That's a cycle. It's a consistent cycle. There's no variation in that. Here, this is just an image to show, here's a grass root. This is actually a Poa annua. This is one of our experimental plants that we use. You can see here at the tip, you can see this little, like a little halo around it. Those are bacteria. Those are bacteria that the plant root is cultivating around that meristem. And it is taking in those bacteria into the cells right there at that tip. Okay, and this shows where the root hairs are here. You can see the root hairs there, and you can see the bacteria coming out. This is fluorescent. They're stained with a fluorescent probe for basically bacterial DNA, but it shows where they're ejected. They're ejected. They come right out of the hair tips every time there's a growth. So you get a few bacteria enter at the root tip. Many bacteria are produced and ejected back to the soil. So these plants are really increasing the microbes that work with it in the soil, in the rhizosphere around these roots. So, and here, look at this. This is actually the third image. This is a root hair. And you can see this is a Poa annua again. And you can see these little spherical things in there. Those are the bacteria, the protoplasts inside. And you can see coming out again. You can see them reforming their cell walls and coming out. So This is a root tip showing where, this is a grass Phragmites, showing where actually the plant is cultivating the bacteria. And so this is where the exudates are. And you can see most of the bacteria right here near the tip. And you see a few, they go fewer and fewer. That's as the nutrients go out. So higher in concentration here, lots of microbes being cultivated there. They're taken in. This is actually a branch root. So it's just a very young branch root showing where this plant is absorbing microbes into the root tip. This is a root tip that, again, Poa annua. We like these little grasses because they fit under a microscope slide. So this is stained with a bacterial stain called crystal violet. And this is the tip here, and you can see in here these little purple things. Those are the bacteria when they're first taken in. That's what they look like when they're first taken in. They have their cell walls there. If you go past the tip, you go back here, this zone back here, it's a bit lighter in color. This is where the plant begins to extract nutrients from these microbes using superoxide. And what's happening here is that these cell walls on the bacteria are being oxidized off. They're basically falling off of the bacteria into these root cells. So whatever nutrients were on those bacteria, on those walls, the plant is getting right there in that zone. If we look these insets, you can look to the right, you can see that's just from the tip, and you can see spherical they are, and you can see how dark the wall is. The cell walls are still there when they first go in. But over here to the left, you see they're irregular. Those are the walls have fallen off and already have been oxidized off. And this is a close-up of that. You can see the irregularness now of the dark crystal violet stains bacterial cell walls, and it stains them that dark. There's little pink things. Those pink things in this case are where the bacteria are, but they have no cell walls. So the pink, and you don't see them very much because they're getting obscured, but you can see them here and there, the pink. And so those are without the cell walls. So the dark is the cell walls that are falling off in the plant. The plant takes that. The plant it's like really taking sheep, right? Taking sheep. It's like ranching instead of farming now. The plant brings these bacteria in, puts them in the cells, and then shears off their cell walls. Okay, and so in this respect, it's like taking sheep, shearing the wool, and then putting them back into the soil. So this is just I don't know if you can see the paper, but this is actually from a paper, from an article by Thomas and Rice in 2014, actually discussing how nutrients are carried on the cell walls of bacteria. And many of these metals are actually, divalent metals actually come into plants on the walls, on the cell walls of bacteria. And, the physics of that, or I don't know exactly what the physics of that is, but this is an image that shows these little dots, and I think those are silver particles on those bacterial cell walls. So bacteria carry nutrients on their walls, and even if the plant doesn't oxidize them fully and fully degrade the microbe, which many are not, many cycle all the way through, the plant can get nutrients from those microbes. Okay, so let's go to this is what these protoplasts look like now. This is a root cell, root epidermal cell, and it's stained for hydrogen peroxide, one of the reactive oxygen stains that I talked about. This one stains hydrogen peroxide. It's a stain called we called it DAB, diamino benzidine tetrahydrochloride, diaminobenzidine, DAB. You can see it up there, acronym, but it stains hydrogen peroxide, and you can see these bacteria brown. That's because of the hydrogen peroxide accumulating in them and around them. Okay, because the plant is putting superoxide on them, it's causing hydrogen peroxide actually is forming in that process. But what you can see is there's a little chain there. You can see the smallest bacterium there, the smallest, and you see it's blue. That blue is protein. We use aniline blue, which stands for protein. So you can see the little one has all its proteins. You see the older one, the bigger one, is losing protein. That is because what happens with these, with superoxide, because the plant is secreting superoxide, putting them onto the microbe, the superoxide is very potent and can go through the bacterial membrane. And these are the protoplasts. So there's a membrane there, very little wall material, if any, but the superoxide can go in and it clips up the protein in these older cells. So inside the plant cell, the microbes are constantly reproducing, constantly trying to stay ahead of destruction by superoxide and full degradation. So hence, and in fact, plants are actually increasing these protoplasts inside them. These protoplasts, the plant, because these protoplasts don't have cell walls, they divide real fast. So the plant can make a lot of copies of these microbes that are inside it, these protoplasts inside it. And so the plant can make many versions, clones essentially of these. And then all these clones of microbes that the plant makes, it can then put out back into the soil from the root hairs. And I'll show you how that. This plant is Phragmites. That's one of the plants we work on. We have funding from the U.S. Geological Survey to figure out how we can control weeds using microbiology. And so this is where some of our work comes from in understanding that process. So anyways, that's why we're working on this Phragmites, this giant weed, an invasive plant in North America. This is some ultrastructure that is of a root. It happens to be an amaranthus, an amaranth, right? Amaranthus, a flower. This is the seedling of the root and a root cell here. And what you can see here is that you can see here, this is in the cytoplasm. You can see this is a mitochondrion here, MM. And you see this thing here. This is a bacillus. This is a bacterium without its cell wall. It's inside the cytoplasm in this case. Here we have again in the cytoplasm, it's a bacillus, and here we can see an endospore forming in that bacillus. So these bacteria go even in the cytoplasm. We didn't think that initially, but it seems pretty clear that that's going on. So this is a diagram of a root cell. The gray is the wall. In this case, the membrane here, the little line, that's the cytoplasmic membrane. These bacteria initially go underneath the wall. They're absorbed into the cells. They're taken into the cells. We don't know exactly how they're taken in. There are a couple of ideas. I won't go into them because we don't have enough proof to really back up either one of those mechanisms or any of those potential mechanisms. We can see them going through the cell walls. We don't know what the process is to take them in, whether it's charges or some other process to get them in. We don't know. But they're taken in through the wall because that wall in these dividing cells or recently divided cells is not hardened yet, so they can go right in. And then the bacteria are detected by the plant, and the plant cell puts superoxide. This 2O-, that's the superoxide. And superoxide is highly potent. It comes from oxygen. It basically where the plant, and I'll show you how that happens, where the plant and the enzymes that are involved in that, and the enzyme, the most important enzyme that does that, takes the oxygen from the air, turns it into superoxide, and then uses it to extract nutrients from these bacteria. So, and the other process that the plant is doing that you probably have never seen in roots before is cyclosis. And what the plant root cell will do, it will move these microbes around in a circular fashion. And I don't know if there's more one way than the other, but we see this all the time, that these roots, especially in the root hairs, are cycling the microbes around the length of the root hairs. And we think that's a way to continue extracting nutrients from the microbes, but it's also a way, probably more important, to make copies and break up these microbes so that the plant can get more and more. And I'll show you how we think that happens. This process is cyclosis, termed cyclosis. You don't see cyclosis in root hairs if you pull root hairs out of the soil. And the reason you don't see them is because cyclosis is dependent on the cytoskeleton of the cell, of the root hair. And if you pull the hair out, you bend the root hair and you break the cytoskeleton and you break the capacity to move those microbes around. So you won't see that. But I'll show you an image of that. I'll show you actually what that looks like. So the enzyme that produces superoxide is this one. And it is one called NADPH oxidase, otherwise known as NOx. It's in the plasma membrane of the root cell. It's in the plant root cell, in their membranes, maybe even deeper in endoplasmic reticulum. I'm not exactly sure where else it is, but we know it's in the membrane, in the plasma membrane. And what happens with this enzyme is that it will take oxygen from the air, and of course this oxygen has to be in the soil, has to be in the air spaces in the soil. So the root hairs can actually get this oxygen. Roots can get them from the air spaces in the soil, and then take that oxygen and turn it into superoxide, and then use that superoxide to extract nutrients from those bacteria. If for some reason plants are unable to get this oxygen from the soil, say for example, if soils are flooded, then one of the effects is that plants cannot make superoxide, and then plants cannot extract nutrients from bacteria that they've absorbed into their cells. And so plants will suffer because of that. The other effect is that these microbes actually in root cells are secreting ethylene. And the superoxide, and that is a growth hormone, and I'll explain that. That's a growth hormone, but it also is a stress hormone. And if the plant can't get that superoxide from the air, then it cannot control the microbes, and the ethylene builds up, and that ethylene causes stress in the plants, in the plant roots. And so we know that plant roots that can't get oxygen, say in flooded environments, are high in ethylene, and they also produce very limited amount of nutrients. It limits the nutrients, and so plants really suffer when they can't get that oxygen in their roots. And we think it's this process because of this, when this process doesn't work right. Another thing I want to point out is that this enzyme uses iron as a cofactor. So one has to be careful that plants are able to get this iron. So one should minimize, especially in soil around plants, should minimize any kind of chelators that would absorb iron and make it so that plants can't get them. Of course, without iron, plants are going to suffer in a lot of ways. But one way is this: they cannot then create superoxide. They can't do efficient rhizophagy cycle. They can't get nutrients from the microbes. So this just to show an image. It shows bacteria with their cell walls. They may be rods. With the superoxide oxidizing off the cell walls, they become these little protoplasts. This is nothing. These protoplasts are wall-less. They divide real fast. They're different sizes. This is nothing new. This is not anything that we invented in my lab. And I know it seems kind of crazy that this is happening, but this is long known. This was actually discovered at the Lister Institute back in the 50s or 60s, or maybe before. Maybe it was before. I don't remember when it was discovered. But in medicine, medical scientists discovered these L forms first, coming out of animals. And then in the 1970s, plant pathologists were studying this process, and they were seeing these L forms too. And then it kind of died down until we picked it up again more recently. But these protoplasts, because they were discovered at the Lister Institute, they become named as L forms, and that L is for Lister Institute. So just to just to put the details, some more details there, so you see. And you can look up this. There's many papers about this stuff. So this is not, I'm not talking about things that are new, that are made up. If you put your mind to it, you get into the literature, you'll see where all this comes from. So it turns out that microbes in plant tissues, in roots, doing rhizophagy, the plant rhizophagy cycle is really important for plant development. If we take away all the microbes from a plant, seedlings we can actually sterilize seedlings and remove most of the microbes. And so when we do that, then plant development is messed up. In other words, plants have to have these bacteria inside their cells to develop properly, especially roots. Roots will not develop well at all without bacteria inside their cells. And for example, and I've alluded to this already, the microbes themselves inside the root cell are what causes root hairs to grow. If we remove the microbes, we have no root hair growth, as in no root hair growth. They will not elongate. They will stay there. You'll have a root hair initial that will not elongate unless there's bacteria in there. Now, why didn't we discover this before? Because we didn't see it, because we just couldn't see it, because bacteria aren't supposed to be there. Okay, but they are. Also, another effect is that these microbes on roots actually trigger the gravitropic response. When we remove microbes from plants, especially grasses, where most of our experiments are with grasses, but also other plants, including some tree crops where we've removed the microbes off of trees, what happens is the roots themselves don't grow down like they're supposed to. Instead, some roots will grow into the air. Most roots will just grow on the surface. Occasionally, some will go down, but they do not go down like they're supposed to. We don't know what causes that. I recently have seen a paper that suggests that it's a hydrogen peroxide effect. I also saw another paper that suggests it's ethylene involved, and both of those are involved in this interaction with this microbe. So that would explain why the microbes have to be there. Also, it's well known that root architecture is determined by the microbes around those roots, and that is, you get more root branching, you get more root exudation, exudates going out when the microbes are there, more branching, more root growth. Basically, a healthy root is going to, with lots of microbes, will be heavily branched, have lots of exudates, and a bit bigger, very different than chemically fertilized roots. And then there is an effect on shoots, because those microbes are mobilized up to the shoots. And of course, if you have limitations of process of nutrients and microbiology in the roots, there is going to be, with reduced root growth, there's going to be reduced shoot growth. But the biggest impacts are going to be below ground. You're going to see huge differences when you remove microbes off of seedlings. And again, you don't we don't sterilize a seedling, we sterilize the seed, right? We sterilize that. Just so you know, full disclosure, all we do is take the seed and we wash it in Clorox, 50%, basically Clorox diluted half and half with water. And then we agitate, we wash for like 50 minutes to an hour to remove all those microbes. Then we rinse all that Clorox off, and then we germinate the seed. And then we either put microbes on it or we don't. And I'll show you experiments where we've done that. Oh, here's something. This is a Bermuda grass root. And you can see the root tip out here. What we did here is we sterilized the Bermuda grass using the Clorox, using the process I talked about with using Clorox to clean the seed. And we germinated it, and you see the root, beautiful root. You see it's black there. That's because we used hydrogen peroxide on it. I mean, the H2O2, the hydrogen peroxide stain on it, and it turns it black like that. So you see there's H2O2 around the meristem. But you see back here, you don't see much H2O2, but you can see the little bumps. Those little bumps, those are the root hair initials. They don't have bacteria in them. They don't elongate. But in the same experiment, we took seeds, removed the microbe, same thing, just like this, and we put a microbe on it, a microbe that we pulled out of that invasive Phragmites. But we did this with lots of microbes and put them on, and we see the same thing. This is what you see. Put it on, and the seedling comes out, and now you see the root hairs are elongating. And you can see, if you see the brown there, that's the bacteria in there. This is the hydrogen peroxide stain. You can see them with the hydrogen peroxide. And if we look at older part, look at this. Here's older part of the root. You can see all the hairs, all the brown. Those are bacteria inside. Those hairs, all the little brown dots, those are all the bacteria inside there. These hairs, these root hairs, they're about these microbes. Are they about absorbing nutrients from the soil? Well, probably. That makes sense. That makes a lot of sense. But they're really about processing microbes and putting those microbes back out into the soil, the surviving ones, repopulating the rhizosphere with these microbes. And we think that because we don't get the hairs without the microbes. And the other reason is that these root hairs don't really live much longer after all these microbes are gone. After the microbes are ejected, most of these root hairs will dry up. And most of a root doesn't have root hairs on it. It's root hairless. It's hairless. The root hairs are only down at the tips. They're only at the root tips. That's where Rhizophagy cycle is happening. So all arrows point to a microbial processing function to root hairs. This shows a root hair. This is from that same plant. This is Bermuda grass. You can see how many of these protoplasts, these bacterial protoplasts, are inside there. They're in there, and this is stained again. They're brown because it's stained with hydrogen peroxide stain because of the reactive oxygen interaction with the plant. So why do these bacteria trigger root hair elongation? We've done a series of experiments, a number of studies that have shown or that have suggested that the reason that elongation happens is because the microbes are secreting growth substances, growth hormones, growth promotional substances, hormone. Ethylene is one of those, and we can detect ethylene using our histochemical stains for ethylene, and we can see them around the bacteria. And that is a known growth hormone, known that growth in root hairs has been related to growth ethylene production. The other chemical that we detect is nitric oxide, and that's another one that we can detect in there. The nitric oxide is also a growth hormone in plants, but we also can detect nitrate in there. And we don't know if the microbes themselves are secreting nitrate. Well, they could be secreting any form. We think they're secreting multiple forms of nitrogen, and then those forms are being oxidized and nitrate is being formed. But nitrate also causes growth, but it's endogenously formed inside the root cells. Okay, so of course we use nitrate as a fertilizer, but this is very different because it's endogenous. It's inside the cell that we see that formation. This paper was, again, this is if you want to look at some of the work that supports what I'm talking about, causing root hair growth and the microprocessing root hairs, it's this paper. Chang et al. Chemical interactions at the interface of plant root hair cells and intracellular bacteria, published in Microorganisms in 2021. And this actually is I'm going to show you now how plants are processing these bacteria inside their hairs. This is a sedge that occurs actually in the island of Bonaire. Number of years ago, I was able to go there and do research. And I studied this. One of the plants I studied there was this little sedge growing on these rocks in these limestone, these coral fossil deposits, right? And they grow in pits. They don't have soil, they have sand, but they have lots of microbes inside them. And they have lots of microbes that they're processing. And I'll show you that. That's why this is kind of interesting. Okay, if you look, this is a root hair. sedge. And you can see in it now, you can see where the bacteria are, because you can see the brown there. That is the hydrogen peroxide. That's where the superoxide is coming from the plant put onto that microbe. The blue, the slight blue, I don't know if you can see the blue, those are the bacteria. That's the aniline blue again that I put in there. You can see it's light blue. But this is stained. This is dead, right? This is stained. Stained, no action. But if you look above now, this is living root hair. This is what few people have seen with their own eyes. And you can only see this if you don't move the hair. And we can see this because we leave the root growing in agarose. We put in the agarose so that they go to the bottom of the plate, to a plastic clear Petri dish plate. And then we turn that over and we visualize that with a microscope now. And then we can see what really is happening. And you can see that these hairs are moving these microbes around. These shadows that are moving, they are the bacterial masses that the plant is processing. The root hair is processing these microbes. That's breaking these bacteria up, these protoplasts, making copies of them, increasing their numbers. It is, and we think also that for bacteria that can fix nitrogen, this is also a way that some nitrogen can get fixed, because the plant, in breaking these microbes up, forces the microbes to engage their nitrogen fixation process and refill their cells with new protein. And so then as the plant degrades those bacteria, it can get that protein, right? So it is a possible way that plants are getting nitrogen, more nitrogen, again, endogenously. And it's not just here, it's all over the plant, in the leaves, in the fruits, in the roots. We see these kind of interactions happening. It's endogenous. But most of the extraction is actually happening in the roots. And so the roots are rich in microbiology. They are the richest part of the plant. But some of those microbes, the plant does move up into the leaves and onto the fruits and seeds. And, but anyways, that I wanted to show you that so you can see that. This shows actually what these root hairs look like. And these are the bacteria in it. And you can see they're clusters of bacteria. And these are the protoplasts. And you can see there's big ones and little ones. Of course, the little ones are the youngest ones, and the big ones, some of these look like they're getting lighter. They're getting lighter because, like this over here, they're losing their nutrients. They're being extracted. You see some that aren't very dark at all. They're breaking down. So the plant is breaking these cells down inside and replicating them, making lots of copies. And that's important because the plant is going to be putting these microbes out into the soil. And these are the microbes that respond to the plant, that come when the plant calls with exudates, and the plant can take in and get nutrients out of them. So these are the partner microbes. Some microbes don't respond, and so those don't go, but the ones that respond come in. The ones that are ideal actually then go through, deliver nutrients, and then they're pushed back out to the soil, right? They cycle through, and those are the ones the plant replicates. Those are the ones the plant builds up in the rhizosphere, so that they can use for growth. This is actually a root hair, living root hair, and you can see what's happened here. You can see the plant root hair is moving the microbes, all these white things, to the tip, and then they're accumulating here in the tip. You see them bumbling around, a bunch of them bumbling around like little ghosts, little bacterial ghosts there at the tip, bumbling around. Every now and then we can see one that gets caught. You can see they get caught back in, and you can see they get pulled back. But most of these in bulk just stay there, right? But every now and then one gets like that when you can see it going right to there, right there, gets pulled back. So, but this process, what we think is happening, okay, the plant is moving the microbes to the tip. They're accumulating at the tip. They're bumbling around at the tip. Then they, in bulk, in mass, will secrete ethylene. And that ethylene, they'll also secrete nitric oxide. And I'm not going to go into the details about that, but I did refer to papers and the papers where you can go look at that. But nitric oxide and nitrate are also involved. So nitrogen and ethylene is involved. And then that causes this root hair to grow. And that root hair will grow. It will have a growth spurt. And when it grows, these bacteria that are in the tip will be ejected out of little pores because they're protoplasts. And so just little breaks in the tip of that hair, those protoplasts will squeeze out of that when that hair grows. And then there's some in the edges, these ones that are in the edges that we see moving around, those will be the ones that are left. And then they'll accumulate like this, they'll move like this, and they'll go, and I'll show you a diagram that shows that, but they'll accumulate in the tip, they'll replicate them in here, they'll accumulate in mass, and then again, in mass, they'll secrete those growth hormones, and then you'll have another growth spurt. It's really a cool process. Well, this is one of these root hairs. This is one of the sedge root hairs again. You can see how fat it is. These root hairs, you look at all kinds of plants, you'll see this. You go and look yourself, get a microscope, and you'll see these things. You'll look at these hairs, but you can see this, how fat this hair is. You can see the bacteria here that are stained with hydrogen, the DAB, the hydrogen peroxide stain, but all in here, all of this cottony white color, that's all bacteria. They're all still bacteria in there. These hairs become fat with bacteria. And they're swollen because they're also very plastic or soft. And so they can go out, and you don't see pores here, but I'll show you some. I'll show you ejections, actual ejections. You can see what out here from the tip, this is where the bacteria came out. And they come out here, and you can see some of them are rods, because what happens out here, once they're ejected and they're no longer exposed to the superoxide, they're no longer exposed to reactive oxygen, they've reformed their cell walls. This is where the plant root hair, when they eject the microbes, eject some nutrients with those microbes from the hair tip to rebuild these bacteria. So if they have cell walls, well, they'll rebuild their cell walls. If they have flagella to swim, they'll rebuild their flagella, and they'll go back out into the soil, acquiring more nutrients, and later to be attracted back to the plant. This is an image that shows a tip. This is, I think this is a basal seedling root. I believe it's a root hair, and you can see the blue here, it's stained differently. You can see the bacteria in there, and you can see here, there's actually pores and it looks like those pores have some bacteria that are accumulating there. You can see, actually, you can see little lines there. Those are pores in that wall. And so where the bacteria come through, this is a mass of bacteria that came out of that root hair tip. And this is just after ejection, but I'm just showing you a couple of different images there so you see. And this is that process that I'm showing you. This is a diagram. This is the accumulate microbes, root hair tip, cyclosis, moving bacteria to the tip, hormone secretion, causing growth spurt of the hair, ejections through little pores, bacteria get ejected. The ones that remain here then are processed, they're replicated, increased, they accumulate, they're taken to the tip, they accumulate, and again, hormones produced by the microbes and an ejection like that. And that keeps happening. And the root hair grows. That's how root hairs grow. Root hairs don't grow any other way. I don't care what we've learned before. We don't see root hairs growing without bacteria being inside the root hair itself. They don't grow any other way, despite all the data that we have, all of our suppositions. It doesn't seem to work like that. They have to have bacteria in them. This is root hair, and it is actually a Bermuda grass root hair. And in this plant, we took a bacterium from a Japanese plant called Japanese knotweed, another invasive. Again, we work on invasives. We're working on some invasives too. And so we got a microbe, an endophyte from this invasive plant, and we put on Phragmites. And it's obviously, it's not even, the Japanese knotweed is a dicot. You know, this is a monocot. So you wouldn't expect they would be totally adaptable. And in fact, they're not. Because what happened here is that the microbe goes in as an endophyte. It's taken into the root cells, and they trigger root hair elongation, but they're not ejected. They stay in. And so you can see here these little marks here, these little dark areas. Those are where the bacteria were not ejected out. They stayed in. And what happens, let me see if I can see. I'll look here, kind of here. You can see where my arrow is. You can see there's bacteria that were there. This was at the tip. Those bacteria stuck, and the growth happened around them. And so you see it kind of turns, and you have this clear area. That's where the growth spurt was. And then you see just a bit later, you see more bacteria accumulating there. And then you see that there's another growth spurt. Those bacteria stayed in there. They weren't ejected properly. You saw there was a growth spurt, and then you see some bacteria there. They weren't ejected. A growth spurt like that. Like that. Here, the ultimate one, you can see the bacteria that were in there that caused the growth spurt, but you can see they didn't get ejected. They remained in, and there you can see the growth spurt happening of the hair. Sometimes we see root hairs that will grow like this, and then they'll turn, grow like that, and we'll see that the root hair got plugged with bacteria. There's a reason it turned, and then it'll get plugged again, and then it'll grow from the side again like that, back and forth, back and forth, back and forth. That's because the bacteria are involved in that growth process. We also sometimes see bacteria, root hairs that will make Y, that'll divide in a Y. And what do we see there? We see that there's a big wad of bacteria plugging up the root hair right there, right in the middle. And so the hair divides like that. I've seen papers where they say that's genetic, but it appears not to be. That this is microbial related phenomena that we're talking about, endophyte related phenomena. This is tomato. This is my student, undergraduate, who worked on that. And what you can see here is the bacteria in the tomato root hair. And if you look at 3, this is the actual ejection process. If you look down there at that arrow right at the tip, you could see there are, at least I can see it. I don't know if you could see it. There are 3 little pores. And those bacteria that were in the tip are being ejected through those pores out here. And of course, that's where they will reform their walls, their cell walls, and flagella if they have them, and go back into the soil. That is how root hairs work. That's what's happening. This, it was hypothesized early on that this is actually the Rhizophagy cycle. Rhizophagy is a nutrient absorption mechanism. This is, but it's also developmental. There's a lot of things that happen because plants absorb these microbes, but it appears that nutrients are critical in a critical outcome of this process. And it's not easy to figure out exactly what nutrients. There's a lot of complexity going on there. But basically we know this about plants and where their nutrients are coming from. We know that plants can acquire nutrients in the soil water, solubilized from soil water. We've thought that for hundreds of years, a couple of hundred years, I think. That plants will just suck those nutrients right out of the water, and that's why our chemistry works. That's the reason. That's the only reason why our chemistry works. Otherwise, our chemistry wouldn't work. We also know, and we've known for decades, and have hypothesized for longer than that plants can also get nutrients from mycorrhizae. Mycorrhizae, fungi that plants affiliate with in the soil, and then use the mycorrhizae to acquire nutrients. We know that. And there are nutrients now which we know come from oxidizing bacteria and other microbes that are absorbed into the root cells in the Rhizophagy cycle. And separating out the sources of which nutrients come from soil water, which ones come from oxidized, which ones come from mycorrhizae, is tricky, especially these first two, figuring out where those nutrients come from, what comes from what, okay? What we have done to try to answer the question of nutrient absorption from these microbes is a number of different kinds of experiments. One kind is isotope tracking experiments, and I'll explain. I'll show you how that happened. And then the other things that we've done is we have grown plants with and without microbes, or removed most of the microbes just by seed sterilization, and then we left microbes on it, or we've removed microbes from both and then put microbes back in one set, put microbes back in seeds in the plants, and then left them off of other plants, and then measured nutrients that went into the plants. Those, when we do that, we see huge differences. When the microbes are there, plants thrive. They have lots of nutrients. When they're not, they're starving. They're sick. They'll get diseased. They're puny. When we analyze the nutrients, they're very, very low in soil available nutrients. And then the other thing that we've done is we have tried to suppress oxygen using carbon dioxide, and I'll explain that. And then we try to look at the difference between nutrients plants can get with oxygen, and then nutrients that plants get when we suppress the oxygen, the superoxide, right? So basically we suppress the oxidative process. And one experiment that we did is kind of interesting, an early experiment where we labeled some bacterial DNA with actually with nitrogen-15. So we just grew the bacteria in nitrogen-15. They absorbed the nitrogen, the isotopic nitrogen, the 15 in nitrogen, right? The heavy nitrogen isotope. They absorb it, they put it in their proteins. Then we extract all the proteins and we put it into agar media, just agarose. And then we grow plants on that. And then we see how that works. And this just shows kind of what we do. And here you can see, here you see two treatments. You see what we do, how that experiment looks. This agarose actually has some bacterial labeled 15N labeled protein in it. And this agar also has the 15N labeled protein in it. But the one to the left has bacteria on the seedling, and the one to the right lacks bacteria, or bacteria were reduced in the seedling. And what you can, well, you don't notice anything now. You can see actually the red here. The red is a hydrogen peroxide, because we stained this with hydrogen peroxide to show. So you see there's more hydrogen peroxide around those roots where the bacteria are than without them. Okay, so that again, indication of the interaction there, but that's why that's where that's coming from. This is data that shows, and this is very interesting data set. But basically what it shows is that in the situations where what we did was, now we cut the leaves off and we analyzed the leaves for nitrogen absorption, nitrogen-15 absorption. And you see when we had bacteria there, you get this much nitrogen-15 that was absorbed. And when we lack the bacteria, or we reduced the bacteria, then you get this much absorption of the nitrogen-15. So it's about a 30% difference. And one curious, interesting outcome, these were all Petri dish plates used in a same canister. And if you look here, this was one of our controls. This was no protein in the agarose. This was no bacteria. And when we analyzed, we got the seedlings, we analyzed them for nitrogen-15, we got very little, nothing there. There's no bacteria there. Okay, but here in another treatment, we had no protein, so there's no nitrogen-15 in the agarose there. There are bacteria there, But look, there we did detect nitrogen 15. What happened? What had to happen is that there are gases coming off of these other plants that from the nitrogen 15, there may be nitric oxide or some nitrogen gas that comes up. And then these plants with their bacteria in them can acquire that nitrogen from the air. From the air. That's how efficient they are with the microbes. They can get them from the air and then the plant can get it. So the microbes are critical in nitrogen accumulation and nitrogen use efficiency. This is data, a microarray study from my graduate student. Her picture is there, Dr. Eve Irizarry. She's now a professor in Puerto Rico at one of the universities. She's moved from two or three universities there. But she looked at cotton. And with cotton, what she did is she got a cotton microbe out of wild cotton or a cotton relative, from Puerto Rico, actually. There's a big tree. I forgot what it's called, but it's a cool tree. And it's a cotton family. Took the microbes there and then happened to be a Bacillus amyloliquefaciens. And then she took cotton plants, seedlings, and then she inoculated some with this endophyte from this wild cotton tree. And then she compared the two in terms of the pathways that were stimulated by the microbe. And what you can see here, these are the top pathways here that were stimulated when the microbe was there. And you see nitrate metabolism process, you see nitrate assimilation process. You see also cellular nitrogen compound metabolic process. So all these nitrogen, because these microbes are there, all this nitrogen, again, nitrogen use efficiency. You see other things going on like heme binding, metal binding. So iron is important in rhizophagy process. It's important. The heme binding, copper ion binding. I don't know why that's important. I don't know why that's there. But we have these other membrane-bound vesicles, microtubule cytoskeleton. The process that the cytoskeleton, the cyclosis, that also microtubules, microtubule-based movement. Look there, that was upregulated. All that is rhizophagy process. And nitrogen use efficiency. This is what we're talking about. This is why those microbes are important. Even if they're not nitrogen fixing microbes, they're important in nitrogen use efficiency. So we also did experiments where we suppress superoxide in seedlings, and then we try to measure the nutrients that come from when we suppress the superoxide compared to when we don't. And we look at the difference, and I'll show you how we do that. But what we do basically for these experiments is we put a lot of carbon dioxide in a chamber and we grow plants in that chamber. This is not just a little bit of carbon dioxide. This is not at like 1,000 or 1,200 part per million CO2, and like we do in greenhouses when we push tomatoes or crops in the greenhouse, right? We just put a little bit more CO2 in there. This is a lot of CO2 to suppress the superoxide. It takes a lot. So, I mean, it's a big number here. It's a big number. Basically, it's just we're using dry ice to get them to go in. We're just using dry ice, and we're sublimating that dry ice in these chambers. And then we're measuring how much it is to approximate the level here. It's a lot. It's a lot. Okay, and this is basically from one of these kind of experiments that to kind of show that actually this is in Petri dishes, but the most of the experiments we do are not in Petri dishes. With this experiment, they're in potting mix, they're in soils, but just the chamber, the kind of chamber effect. This shows the suppression of the superoxide with the CO2 and how that works. To the right, this is wheat, and this is stained for this purple is superoxide. So we use the stain. It's called nitro blue tetrazolium that turns purple when there's superoxide there. And so you see the superoxide is all in this tissue. That's just in the air. The plant could get oxygen. This to the left is a same experiment. This is high elevated carbon dioxide, stained again for superoxide, but you can see there's no purple around the edges where the carbon dioxide went. Now there is some in the center because the carbon dioxide didn't reach in the center, but the microbes are really not in the center. The microbes are out here. That's where the plant is processing those microbes. So we suppress Rhizophagy cycle using this elevated carbon dioxide. We're still using this technique because it's really valuable. We're still trying to figure out, all the nutrients that are involved, but I'll show you data. Okay, so basically, to calculate from these kind of experiments, to calculate the not rhizophagy specific or oxidation specific nutrients, it's this. And this doesn't have to do with the nitrogen fixation process that may be happening and getting nitrogen out, but specifically, anyways, I'll explain this. This is the oxidized, the ones that are nutrients that are critical through oxidation. And I'll also mention this. From our experiments, I can tell you that every single microbe is different in terms of the nutrients they provide. So plants take in communities of microbes from the soil, and they're getting all kinds of different nutrients because they're taking in multiple kinds of microbes, multiple species of microbes. They're all different. Microbes carry nutrients. It's almost like a fingerprint. Now, why exactly, I don't know, but anyways, this shows, okay, this is an experiment. Okay, look to the see data. We used 4 different bacteria here, and we use the natives. Just look at the native bacteria. Native bacteria, that means we use the bacteria that were just on the seeds. When you look at this, you can see, okay, so here the blue shows the amount of, in this case, we measured manganese in the plant, manganese in the plant. And you can see you get this much manganese in the air, in regular air with oxygen, the amount of oxygen. So this is with oxidation with rhizophagy, right? This much. And if you look at the seedlings or the plants, then where we suppressed the superoxide using CO2, you get this much manganese. It falls precipitously. Okay, these other bacteria, you see they're different. They're all a little bit different. These are all bacillus, different bacillus species that we were doing these experiments with, that we were playing with. This is iron. And with iron, you can see the same thing. With iron, you see, with oxygen, you see a little more. You see when we suppress that, you see less, right? And some bacteria show more of a difference. I mean, look here, Bacillus 2 seems to be really, really good in the oxygen. You seem to get this much iron, and when you suppress it, you get half of it, right? So it's more important for this bacterium. More iron apparently is coming out of this bacterium than it is. That's how we interpret that. So we do that for lots of nutrients, right? Looking at multiple nutrients. And when we do that, what seems important is that these divalent metals seem really important for oxidative extraction. I mean, divalent metals, manganese and iron, calcium, magnesium, sulfur even. We've had other experiments where potassium, we've done experiments with tomato where potassium falls tremendously in the plant if we suppress the oxidative extraction. So it's possible that other plants, that multiple plants are doing it a different way. Some plants show phosphorus as being really effective when we suppress rhizophagy cycle. So anyways, we have a lot to learn about that. And there's much unknown still. We need more people working on this. So if any of you want to work on it, I'll be happy to collaborate on this, help you all as much as we can. But there is another consequence of this process and of rhizophagy cycle and endophytes and the importance of microbiology in plants. And that is that we actually need these microbes to create resilient crops. Plants with microbes are hardier, they're tougher, they're more resistant to stress. They grow better. They need no nutrients added. Plants with microbes. So with the microbiology, we can create resilient crop cultivars for climate change. This leads to some work that was done by some other investigators, Rusty Rodriguez, a good friend of mine, and Regina Redman, also a good friend of mine. A husband-wife team did some work where they showed that plants will associate with microbes that will help them to adapt to the stressful environments. So, for example, if they're growing in salty soil, they'll associate with certain fungi that give them salt tolerance. If they're growing in thermic soils, hot soils, they'll associate with microbes that will improve their resistance to that heat stress. And the paper that describes that is here below, and this is Rodriguez, Henson, other people that describe this process. They called this process habitat adaptive symbiosis, and that is that plants adapt with microbes that enable them to adapt to the stresses in their environment. And the reasons why we think this is happening is that the microbes are actually causing genetic I should say they're causing epigenetic change, possibly genetic change in the host plants. They're actually enhancing the rate of evolution of the plants in addition, but they're also going into the plant and they're affecting the plant's resistance to stresses. And I give you I guess I'll give you an example where plants affect the evolution of the plant where microbes affect the evolution of the plant. And that involves, for example, some of this landrace corn that I'll talk about a little bit about that. But this corn, when it's full of microbes, these corn plants generate much more pollen, bigger tassels, more pollen than plants that don't have the microbes, especially when we compare our modern conventionally grown corn cultivars to some of these landrace corn and hybrids that come from the landrace corn that contain the microbes. You see much more pollen. And when we look at that pollen, and I'm not going to show you this, but the pollen actually has bacteria being vectored inside the pollen and inside the seed. So the corn plants actually will vector their anyways. But I get to talking, it's a little off target sometimes, but the bacteria, because they're inside the plant producing these hormones, nitrate, hormone-like substances, and I call nitrate a hormone-like substance because it's internally produced and it triggers growth and response in the plant, just like ethylene. These two compounds are what's called nucleomodulins. And a nucleomodulin is anything produced by a microbe that affects the nucleus of the plant. And what these compounds, ethylene and nitrate, do is they cause the plant nucleus to divide. They'll cause something that's called endoreduplication. And it's a mouthful, but it's something that we understand. It's understood a little bit now, and there's articles out. I'm not citing an article now, but basically these compounds cause plant DNA, plant chromosomes to divide. And so you'll get bigger cells, and you get bigger cells in part because you have more DNA in those cells. So this is what's happening when the plants have these microbes in them. You're getting this DNA replication. You're getting more DNA replication. If there are jumping genes in there, you're going to get genes jumping around. You get altered expression. You're getting And the interesting thing about it is when you have highly microbial plants, you get this wild look to plants. They're all different. Plants grown with microbes, there's different sizes. They'll have different size trichomes. You grow them with chemical production, chemical fertilization, and they are more similar. Okay, so this is an issue. This is an issue with production, with actually. So, okay, so we have found that the microbes establish such an intimate association with the plants that they actually go into the nuclear envelopes in plant cells, in certain plant cells, like epidermal cells of the plant. This is a potato root cap cell. And you can see the cell here. You can see the nucleus there. You can see these little things here. These are actually bacteria that are encapsulated. They have little sheaths around them, but they are associated with that nucleus. So it turns out they appear to actually be in the nuclear envelope. And I'll show you other images. So this is potato. This is potato. This is a hosta. This is a leaf. In this case, this is a leaf cell in a hosta. And you can see the cell here, and you can see the nucleus here, and you can see these things coming out. Those are bacteria coming out of those nuclei. This is stained for nitrogen, and you see they're actually having some nitrate around them. Are they producing nitrate? You know, what we have not done, which we have not been able to do yet, is to put a gene on the NIF, a reporter gene like GUS or GFP on one of these bacterial NIF genes, and then put that in one of these bacteria that go in the nucleus, and then show nitrogen fixation there. But what we do show is there's nitrogen coming from around there, that they're doing, and the bacteria coming out. And here too, this is ethylene. This is the nucleus here. And the purple is ethylene now. And you can see these bacteria coming out of these nuclei. They actually stream out of these nuclei. They come out. This is part of development. They come from the nucleus. Here's a mint. This is from the leaf of a mint. And you can see the nucleus here. And you can see these little trails of the bacteria. This is trails. The bacteria were in here. Some of them still are in that nucleus. And they come out here, and you can see the bacteria. These are little rods here coming through there, and they stream from the nucleus out to this outside the membrane in the periplasmic space. This is just outside, just underneath the cell wall. So they come out of the nucleus and they go into those areas. And we think then the plant will get nutrients from them in this little, it's called the periplasmic space, by the way, just underneath the cell wall. That when I showed the initial image with the bacteria there, that's where they're going. And so you see these trails. We've even seen images, and we have movies where the bacteria seem to be moving both directions, from the nuclei in and from the nuclei back. So, but these processes are phenomenal, seeing this. But this is an example of uniformity that we see in chemically fertilized plants, chemically grown plants. They're very similar to one another because of the chemistry. With biological plants, there's differences. They're variable. They're more variable. And so that's what I'm trying to show here, this non-uniformity. And we think this non-uniformity is due to different plants absorbing different kinds of microbes, different amounts of microbes, so forth, giving you some plants that are a little bigger, some plants that will have bigger, slightly bigger leaves, some plants that will have bigger hairs, like that. We think that non-uniformity comes from because they're absorbing different microbes from the soil, in addition to the microbes that they may carry with them. So nitrogen use efficiency, and I've already alluded to that. And I'm not claiming that we have super efficient systems like we have in legumes, where the plant constructs a nodule, and in that root nodule, it then juices these bacteria for their nitrogen. I'm not suggesting it. We know it's not efficient like that. We know that this is an endogenous process that goes like this. It causes interaction between the microbe in the plant. And the result is, we think that the microbes are delivering nitrogen. Either they're carrying it from the soil, they're increasing efficiency, or they're fixing nitrogen inside. It's not as much, we don't get as much nitrogen as in legumes, because plants, most non-leguminous plants don't enrich soils in nitrogen the way legumes do. You have luxury nitrogen in legumes. Okay, so much of our science that we've done on nitrogen fixation is in that system. We need new systems to study these kind of systems. So an example of this nitrogen use efficiency is looking, this is a friend of mine, this is Walter Goldstein. He's a corn breeder. He's a corn breeder who's obsessed with nitrogen fixing corn. And so he's trying to breed this nitrogen fixing corn. This actually shows some of his corn lines. Some of these are from Mexico. Some of these are, and he's making hybrids to try to create new corn lines, hybrids with modern cultivars to try to get the agronomic characteristics that we want. But you see how they're grown. They're grown here. This is from his breeding nursery. To the left, you see this is conventional. They look different. Why? Because he's not putting much nitrogen. I understand that Walter puts on 20% of the nitrogen most people are using at certain times. And I can't tell you exactly what Walter Goldstein, the amounts that he's using, but he told me it's 20. It's minimal nitrogen. And so you see the conventional cultivars not performing as much. By the way, Walter Goldstein is he's in Wisconsin. He is at the Mandamin Institute. And he is this is what he does. And so we saw him doing this a few years ago, and I contacted Walter and said, hey, we can help you do that. Of course, I'm not really trying to convert to produce cultivars. I just want to understand how these systems work. So Walter and what he's trying to do is perfect for me to help and then understand how these systems are working. So we looked at these systems. And what we found so far is that in these landrace corn, and you have nitrogen being fixed, at least you have nitrogen. You have microbes, put it this way, I'll put it another way. You have microbes and some evidence of nitrogen fixation in terms of our histochemical stains and in terms of isotopic tracking all over the plant. Most of the nitrogen fixers, by the way, are in the roots. As you would expect, the roots are full of microbes and they're full of nitrogen fixers, but we have also microbes, well, in the roots, in the root hairs, microbes in the leaves, in the epidermal cells, in the trichomes of the leaves. If you have lots of microbes in corn, those corn leaves are going to have really huge trichomes. They're going to have large trichomes. This landrace corn, this nitrogen fixing corn, it'll be hairy. It's hairy. Compared to our conventional corn, there's not much hair. There'll be some hairs, but the leaf blades won't have much hairs on them if they're chemically grown, especially when you use a lot of chemistry. But this landrace corn, they make sometimes those trichomes, those hairs on the leaves will be 5, 6 millimeters tall. They'll be big hairs. Okay, so they're all over the plant, fixing nitrogen. Probably most of it's in the roots, but they're all over the plants. And when we look at this highly efficient, and I'll just call them, I'm not going to call them nitrogen fixing corn, because that comes into the whole thing. And then I have, a lot of people who say, well, the bar for that is really high. You're going to have to then show actual NIF gene expression inside the plant, which we have not done yet. That's going to take somebody who can do something better than I can. But nitrogen use efficiency. And when we look at this landrace corn and the cultivars that come from it, compared to conventional cultivars, and we did a study with Ward Labs in Nebraska, where we looked at actually the fatty acid analysis, comparing the landrace cultivars that Walter was producing, the microbial cultivars, highly microbiological cultivars, to our conventional long history of pure chemical fertilization. And when we do that, what we see is the total compared. Actually, I put the yellow ones here, highlight where we see big differences, where the landrace corn is high. And in fact, any positive number shows the high on landrace corn. And so when you see total bacterial biomass, you see it's really high in the landrace corn compared to the conventional. There's only 3 or 4 cultivars in each of those categories, by the way, so it's not exhaustive. A lot of work, but still, you can see it shows what I'm talking about. When you look at the gram-negative bacterial biomass, you see it's really high in the landrace corn. When you look at the rhizobia, the same bacteria that are in the root nodules, but now they're in the leaves of corn. When you look at that, you see the landrace corn has rhizobia in the leaves, more rhizobia than the landrace corn. Yeah, there's some than the non-landrace and the conventionally grown corn. Okay, so there. Total fungal biomass is even higher, in the landrace corn compared to the conventional. But they're not causing any disease. Why is that? Because there's a whole community of microbes, and this is what's most interesting. When we look at protozoa, the landrace corn leaves contain protozoans. So there's protozoans that apparently are absorbed and moved into the leaves of that. And how there's a whole community of microbes that go into this corn that we don't have in our conventional cultivars. And all of those interactions may be resulting in, between the microbes and the plants, may be resulting in the increased nitrogen efficiency that we see these plants having. And so that increased nitrogen use efficiency, that's an important point. And the other point is, the final point, that these bacteria make plants resistant to diseases. And if we have the bacteria there, we don't need all the fungicides. That doesn't mean we don't need to use fungicides, because not all pathogens are impacted by the bacteria. Some pathogens are not, but I'll explain. And so we'll probably have to have some fungicides. Things like, for example, rust diseases, for example, may not be impacted by the microbiology. But I'm going to show you results of one experiment. This will be the last thing that I'll talk about. But in this experiment, we used basil seeds, and we removed the bacteria from basil seeds. And we compared that to plants with bacteria in terms of how they were resistant to Fusarium oxysporum, a pathogen that causes damping off disease. And this shows this simple little experiment. And when you look at the top 2 plates, what you see the seedlings here. Basically, with these seedlings, to the left, you see seedlings with all the microbes intact. They grew very well. You can see lots of green leaves, lots of root material there. They grew fine. And to the right, you can see what we did was we just took the seeds with their microbes, and we put Fusarium oxysporum in there. And that Fusarium attacked the plant, maybe, or didn't. The point is, there's no disease. There's very little difference. And we did this experiment many times. There's no effect. Fusarium does not impact the seedlings. Sometimes it may colonize the plant, but it doesn't cause disease with those bacteria there, compared to below. Now below, what we did is we took the seeds, we sterilized them with Clorox. Then we washed and removed the Clorox. We basically removed the microbes. We germinated them, and you can see here they're germinated in agarose. You see they're growing. What you don't see is that they have small roots, because the roots don't grow well without the microbes there. Compared to up there, they have big roots. Okay, so the roots were compromised, but there's no problem. They still grew. I mean, yeah, there's a problem, but they still grew. Compared to the situation here, where we removed the microbes, then we gave them fusarium. In this situation, the seedlings, they're cooked, they were consumed, they're destroyed. Yeah, they have to have the bacteria. With the bacteria, they grow fine without it. Disease after disease. A lot of the soil microbes that cause disease don't cause disease when the bacteria are there. This is what we think is going on. We think that the microbes, especially for these fungi, these pathogens that are facultative, that have both a saprotrophic phase and they have a plant disease phase, a pathogenic phase, when those fungi, like fusarium, have the certain microbes, either they're endophytes from the plant or they're native soil microbes, then those microbes will alter the behavior of the fungus, of the pathogen, alter the pathogen so that it's not a pathogen. It becomes a saprophyte, and it grows in the soil as a saprophyte. Doesn't cause disease. Whereas when the bacteria are absent, when those other microbes are absent, then the fungus attacks the plant to get the nutrients that it needs and causes disease. That's what we think. And we can do this. We could show this experiment with a lot of other systems. We've shown it. And I don't know. I guess I show here, basically what I'm showing here is the behavior, the microbe changes behavior of the fungus. And this is actually a fusarium, and this is a bacterium here in the middle. But what happens is the bacterium colonizes the fungus, and the fungus doesn't sporulate. It doesn't sporulate. It grows. It doesn't kill the fungus. It just alters its behavior. And we think it's a symbiosis there that happens. So suppressive soils, rich microbial soils, are suppressive to disease too. Plants with lots of microbes and lots of endophytes are suppressive to pathogens. This is an endophyte, a bacterium that isn't affected, that doesn't change the behavior of the pathogen. And you see it's there, but the pathogen sporulates, it grows. That bacterium doesn't have that effect. So not all bacteria are the same. This just shows the system. To the left is with the bacteria and the fungus there. And you can see the soil is nice and black, and you look to the right. This is no bacteria. You see the seedlings actually are being affected. They're being infected. And you see the mycelium all over the soil. The fungi are growing crazy. This is what's happening in the soil. This is the fungus there, the hypha of the fungus. And you can see the biofilm. You can see the bacteria around it. This is a symbiosis. Those are alive. Those are working together, those microbes. They're not causing disease. That's not a disease organism. That is a symbiotic organism in the soil rather than a pathogen. We can look inside the fungus, and you can see what's happening in many of these fungi. This is alternaria, like Alternaria alternata that causes disease, and in tomato causes early blight, right? Okay, you can see the fungus here, and you see the little green dots. Those are bacteria inside it. The bacteria are actually go inside the fungus. So they're not only on the surface, they're inside. They alter the behavior. They change the behavior. This shows another fungus. You can see the bacteria coming out. You can see them here, the fungus, the bacteria actually coming out there. This is another fungus. This is a zygomycete. It's not a pathogen. Well, some of them are pathogens. Like rhizopus, causing a fruit rot. Fruit rot will grow on fruit, strawberries especially. But you see the hypha of the rhizopus. This is with its fungus, and you can see bacteria coming out. Those are bacteria. They're loaded with bacteria, this rhizopus. Even mushrooms have bacteria in them. They cultivate bacteria inside them, so they need to grow. If you want to grow a healthy mushroom, you don't sterilize it. It has to have its own microbes. But if you look in the mushrooms, take this one for example. If we look at the tissue here, and we look at that with a microscope, and we stain it for nitrogen. This is nitrate stain. This is diphenylamine, acidified diphenylamine. This purple, this blue is actually nitrogen in hyphae, and the little dots there with the little halos around them, those are bacteria inside. Here too, you can see the bacteria. You see some coming out. Those are the bacteria inside. Inside here, this is developing the fruiting body, and this is stained for nitrogen. They make little nodules in that fungus fruiting body where they're getting nitrogen. This is what we say, we'll call it, if we want, we'll call it nitrogen use efficiency. But it looks like the microbes are producing this nitrogen. This is what it looks like. And when we isolate these microbes, they fix nitrogen. They fix nitrogen in culture. They're nitrogen fixers. This shows one of these nodules. You can see the bacteria, the little dots. Again, they're bacteria with their encapsulations. They go in them. So this is something that spans between kingdoms of organisms. I don't know if animals do this, but plants do it, and fungi do it. And the interaction between plants and fungi is partially determined by the fact that they're both doing it. And they can also share microbes. Plants can share the microbes with the fungi. So just to summarize in a minute, and plants are getting nutrients from these microbes. Plants are also, because they're interacting with these endophytes, these microbes in their tissues, plants are becoming resistant to stress. They're increasing their antioxidants because they're using superoxide. They're increasing their antioxidants. So their enzymatic antioxidants are increasing. We've known about this. We did work on fungal endophytes showing this in 2010. Article published. I don't say anything here, but it happens with bacterial endophytes. The same thing is happening. Rhizophagy cycle, this is what's happening. Plants are getting resistant. They're also getting more phenolics. Phenolics are antioxidants, but they also are nutrients. So when we eat plants grown with microbiology, with microbes, they have more phenolics in them. They have more of those healthful chemicals in them that people like. So this increases the chemistry, the health value, and the stress tolerance of the plant. And finally, the microbes in plants and in soils are necessary to control pathogens, control the pathogens. And when we use the microbiology, we don't have to use as much of our fungicides. It's, all of this stuff makes me say that we really want, as a world, as a culture, as a society, as people, we want to move as close as we can to biological agriculture. What I showed initially, growing plants with biology, with microbes, as close as we can. We're pushing crops, we're trying to feed the world, and there may be limitations to how much we can push plants using pure microbiology. So we may be married to chemistry, but we can certainly try to meld the two and produce a cleaner, better agriculture and reduce the damage that we're doing to the environment. This is actually a popular book for gardeners that has been written by a popular writer, a gardening writer named Jeff Lowenfels. And Jeff is also a friend of mine. But he wrote this whole book about Rhizophagy cycle and endophytes. And it's called Teaming with Bacteria, the Organic Gardener's Guide to Endophytic Bacteria and the Rhizophagy Cycle. And so this is on a very popular level. And so Jeff is trying to move this to gardeners and to the common knowledge level. So it's not just something that's at the level of scientists. This is something that gardeners can think about and understand. And it's been received very well. It's only like $25, this book. But Jeff Lowenfels is a science writer, and excellent. He's a non-technical writer. So he puts everything in very common language, very approachable language, which is necessary for people to grasp, I think, easier to grasp than much of our scientific writing. And of course, this work is not my work. This work is done by all of these people. And they're the ones that did pieces, and they're continuing to do pieces. They're postdocs and graduate students and undergraduates. You know, they're the ones that poured their heart out and their energies into getting this stuff, to understanding this stuff. And that is really all of the slides. And I'm going to stop sharing, Moj, and that's all I have to say, Moj. I can't say anything else.
It's great. I really appreciate this presentation. And I think, you know, what you shared with us with regard to Rhizophagy cycle, this should become a part of one of the chapters of mineral nutrition of higher plants by Marshner. You know, that's almost the Bible of a lot of plant nutrition people that study how plants need nutrients. And I think this should be added to the next edition of Marshner's book.
You know, Moj, since you pointed that, I agree. You know, even though we're still trying to understand it, there is a book now that just came out, and I've forgotten what it's titled, but it's Italian. It's an Italian book on soil, on agronomy, soil agronomy, and there's a chapter in it on Rhizophagy cycle. And
That's
so
great.
it's coming out, but it's Italian at this point, right? Italian. Okay, but we do need it in English. We do need it in English. Absolutely.
Yeah, just like when I shared it in our private conversation, that when I go to the agronomy meetings, tri-society meetings, I ask the graduate students if they know about Rhizophagy cycle. These are the agronomy students, and it's kind of a foreign phrase for them. And I'm hoping that what we are doing here will open up the floodgate to the kind of making students hungry for this knowledge. Because if they know it, they are the future, and the professors, crop advisors, if they know this concept very well, they will be able to resolve the drought stress, mineral nutrition stress in plants in a way that is regenerative and it's healthy for environment and healthy for plants. So I have one comment to make, and that is about basically the pathogenic microorganisms become actually, I don't know, not pathogenic. Is that because also when they have the bacteria in them or the microbes in them, is it because those microbes are feeding them so they are not hungry to go after what is the cells of the plants are offering them, and they are just leaving the plant cells alone?
We don't fully know, but that's what we think. That is our hypothesis, that
Okay.
there also is a nutritional interaction between the bacteria and the plant, and that when the fungus, between the bacterium and the fungus, and when the fungus, the fungal pathogen, can get the nutrients it needs from microbes, it never becomes a pathogen. It never goes after the plant to get the nutrients it needs. That's what we think, but it's based on what we see with a lot of the microfungi. Is we could see, like Fusarium, we could see the microbe going in. You know, we don't see necessarily, it's hard to say, well, they're getting nitrogen from them, right? We know they go in and we change behavior, so there's a behavior change. But when we look across the different kinds of fungi, like we go to the mushrooms, and then we look at the bacteria in these mushrooms, in the fruiting bodies, and in the mushroom mycelium, that's where we see all this nitrogen. In fact, there's another phenomenon that's really interesting here. And it is, I call it the wood degradation nitrogen paradox. And it is that as fungi, wood, basically, if you look at wood, the wood itself is low in nitrogen. But as wood degrading fungi, like the ones I showed, like the the reishi mushroom, the Ganoderma fungus that I showed, the mushroom that I showed, as those fungi degrade the wood, the nitrogen content increases in the wood, in the degrading wood. And we think the reason for that is because the fungi are using these bacteria to get the nitrogen that they need to function in that wood, right? So that we think that's what explains that wood nitrogen paradox.
Like the white rod fungi.
Yeah, like
they're
those.
well known for that.
Yeah.
And that's good. Another thing is I know you have touched on the subject of which one is more important, bacteria or fungi in the soil or in plants. And if you'd like to comment on that a little bit and talking about the glomalins and how the structure of the soil can be assisted by either one of these, if you could make a few comments on that, if you don't mind.
I will, but it's a bias. Well, I will tell you first, I am a mycologist and a plant pathologist. You know, we have to wear a lot of different hats, right? We have a lot of different hats, but okay. So I think personally that the bacteria are the most important for plants. I think the bacteria are the most important. However, we do know that you know, and we've known that like glomalin fungi are pretty important in terms mycorrhizae, and pretty important in terms of absorbing, you know, linking up and getting some nutrients like phosphorus, some nitrogen, phosphorus, and some nutrients like zinc and other nutrients too. But it looks to me like bacteria are much more common, much more important, right? Plants don't vector mycorrhizae on their seeds, so they're taking a chance getting those mycorrhizae. But plants do vector these bacteria inside their seed and on their seed. To me, that tells me that the plants are much more reliant on the bacteria. They depend on those bacteria more. They work more with the bacteria than the glomalin fungi. But I will also make a statement about the glomalin fungi. It is also known and may not be appreciated by most people, but it's also known that the glomalin fungi absorb bacteria into their hyphae, and so that interaction happens, and so you get these, you know, these kinds of symbiosis, these levels of symbiotic interaction that involve communities of microbes in plants and in fungi.
Yes, that's great. Well, I really appreciate your time and taking the time to present this, and it's been a wonderful session, and I'm sure the whole science of in agronomy, soil science, and regenerative agriculture will be benefiting from your future findings, as well as confirming and reaffirming your current findings.
Thank you, Moj. I hope I can last much longer, but right now I'm totally exhausted, and I'm looking forward to the weekend. Right now, I have a break. Rest.
You know, you have many presentations recorded and available online in addition to these, and I really appreciate you making one more.
Well, this one is special. First of all, because you asked for this one and you wanted it to have a scientific emphasis, so that agronomists and other scientists could see the data that we have.
Exactly,
and the data. And of course, I didn't show all the data, but I did show examples of the data, and I did include references, because that's where most of the direct data is in those papers that can be referenced by scientists and students interested in knowing more about what we found.
Yes, great. Thank you again.
You're welcome.
And I hope you will have a wonderful weekend.